Hello everyone, welcome back to another class on regeneration biology. In today's class, we will learn about the influence of niches and scaffolds on stem cells and organ culture perspective. So in today's class, what we would focus is mainly two sections, that how and why a niche and scaffold has to play a role or any influence on the cells, and what are the ways in which the cells interact with the scaffold and which can be analyzed and assayed when you are doing a organ culture. So let us see the influence of niches and scaffold on stem cells at first. So stem cell niches and scaffolds regulate stem cell behavior, influencing their self-renewal, differentiation and migration. The scaffolds and stem cells have a strong interconnection, and the niches, as you know, they are immediate neighborhood environment of a given cell is called a niche. Niches are specialized microenvironment that provide physical and biochemical signals to guide stem cell fate decisions. So stem cell fate basically mean if a cell divided into two, one of them will differentiate and one of the two cells newly formed will either contribute to a proliferating group of stem cell, or it will go back to the kitty of the existing cell. So now, depending upon the demand, the stem cells or the number of stem cells present in a group, how many of them have to proliferate. Not that if there is a group of 100 cells, all 100 has to divide. Maybe only 5 will divide. Everything depends on the niche. At the same time, scaffolds act as artificial extracellular matrices that can mimic the natural niche environment that's supporting stem cell growth and differentiation. So matrix and scaffold are one and the same. The word scaffold you use if it is artificially made, and the word matrix you use if it is naturally present. So let us look into the stem cell niches. By definition, niches are specialized microenvironments within the tissue that contain stem cells and cells that support them. Because sometimes in this niche, it may not be just matrix. Sometimes the niche will contain other cells also, who is just like a feeder layer in a stem cell culture, providing these cells that support in the niche. They provide signals that regulate stem cells activity. And if you look into the components, the niches consist of various components that includes niche cells, cells that directly interact with the stem cells, providing signals for their support. Support can be of two types, either maintenance support and also the proliferation support. Then comes the ECM, extracellular matrix. It provides structural support and can influence stem cell adhesion and migration. So stem cells adhesion when needed and migration when cell has to expand into a newer zone. And also secreted factors are present in the niche, that is the molecules that influence stem cell behavior such as growth factors and cytokines. These are all the broad group of secreted factors that influence the stem cells through the niche. And the physical factors, they include tissue stiffness and shear stress, which can affect the stem cell behavior. Sometimes some bending force or a pushing force or some change in the cytoskeletal protein dimension, everything can influence the stem cells behavior, whether to continue to be in the quiescent state or it should continue to divide. Everything is decided by the physical factors also. So now let us see stem cells at home, that is the stem cell niche. Let us see some cartoons. So niche, we have seen it, it's a microenvironment where the stem cells gets a support and they interact with the to and fro interaction. So you can see the niche and the stem cells, which is given blue in color, and they have mutual communication. Stem cell influences the niche and the niche will influence the stem cell. That is this bidirectional arrow. And the direct contact is one influence that stem cell is getting. It is physically in contact with the niche. Either it can be niche cell or the matrix protein. And then the interaction with the niche is through the soluble factors. That can be growth factor, cytokine, as you can see, as you saw here. This is mainly the growth factor and the cytokines, the soluble factors. And then there can be also some intermediate cells. Sometimes, when there is a damage, and the stem cell have to come into picture, and it has to contribute to fixing the damaged tissue by regeneration. Sometimes because of this damage, you will have some intermediate cell that can come into that site, and this intermediate cell is helping in multiple ways. One thing is the communication of the stem cell with this niche will become more effective, more productive. Sometimes this intermediate cell will be able to release some of the chemokines, cytokines, etc. that will stimulate both the niche cells and the stem cells. So sometimes it's just like, friend A and friend B have got some rift. Then friend C comes and patch up the rift or the unhappiness or an argument between A and B. So the presence of C can facilitate this whole process. So this is like a catalyst in a chemical reaction. So this intermediate cell sometimes becomes very influential. They are not part of the niche. That's what we should understand. Many a times they will come from elsewhere and they participate in this interaction of the stem cell with the niche. So now let us see the regulation of stem cell fate. The niches provide cues that influence stem cells self-renewal, differentiation and migration. Self-renewal is making a copy of themselves. Differentiation is a demand-based thing. Whether there is a need for a differentiation, it will do that. Then migration, if it differentiated and sitting there itself, the damaged area will not get fixed. So the cells have to be produced from its source, that is where stem cells are there, and it should migrate to the damaged area so that the damaged area can be fixed. And self-renewal basically means the niches can provide signals that can promote stem cell renewal, allowing them to maintain their identity and generate more stem cell. They have nothing to do with the regeneration, nothing to do with the damage fixing, nothing to do with the differentiation. They are simply making copy of themselves. But remember, stem cells population can be kept in a fixed scenario or fixed state provided you have a healthy population of or healthy number of stem cells, which can be possible only because of self-renewal. Then comes the differentiation. Niches can also direct stem cells to differentiate into specific cell types such as muscle cells and bone cells. So this begs a question, isn't it the stem cells are always interacting with their niche cell, and then why every time stem cell is not differentiating? So the answer is the niche will not send the same set of chemical cues any given time. Depending upon whether the need is to self-renew, such chemical cues will be produced. If the need is to differentiate, such a chemical cue will be produced from the niche. So that's the chemical cues will vary. And then the migration. The niches can guide stem cells migration, ensuring that they are positioned correctly within the tissue. Say a cell is formed and it is not in the correct place where it is supposed to be. So the cells have to migrate properly, and then they will give rise to a well set morphology to the damaged organ. So now what is the impact on the tissue's homeostasis? How the niches will contribute or influence the homeostasis? Homeostasis means running the day-to-day affair. Like homeostasis means like a person got up in the morning, he brushed, had breakfast, those kind of thing, and until then took lunch later and then dinner and they finished the day. So this is a day-to-day event. But one day they may go for watching a movie. That is a change. One day they may go for attending a conference. The day may change. That is not homeostasis. That is different. One day they decided to take a long trip. But if they are daily traveling 50 kilometers, that is homeostasis for them. But if they decided not to travel that day, that is a difference. So this is what you should understand, what is the homeostasis mean. Niches play a crucial role in maintaining tissue homeostasis by regulating stem cells activity. And then comes the stem cells activity mainly contribute to regeneration and also dealing with aging and disease. Regeneration, the niches can be essential for tissue regeneration, providing stem cells for repairing the damaged tissue. This is the main purpose of stem cell and their dynamic interaction with the niche. That is, main purpose is to regenerate, that is fixing a damaged area or a damaged cell type. Aging and disease. Alterations in niche function can contribute to aging and disease development. Many times what happens, like we have discussed, in aged people face you will see wrinkles. Why they have wrinkles there? Because there is too much of wear and tear in certain part of that tissue which is getting fixed, but there is not adequate stem cells available, or even if the adequate stem cells are there, it is not powerful enough, or quantity is not sufficient enough to fix the damaged area. Hence, it is not a wound, it is not a bleeding or a injury site, but the filling doesn't happen, the cellular filling doesn't happen as efficiently as possible. And on top of that, wherever there is wrinkle, the wear and tear is continuing. If wear and tear is stopped completely, maybe and some time maybe it will get fixed. Simple example of wrinkles. So it's not that the stem cells are less there, stem cells are still there, but because of the old age, because of the age, you are not able to supply enough stem cells to fix the damaged area. So this is the simple logic. So aging and same way, sometimes disease, some disease also develop because the tissue is not getting fixed the way it should have been in the young age. So age related diseases come onto various organs. Again, credit goes to the inadequate interaction of stem cell with its niche. Not every disease is because of that, but if you have stem cell, like we have seen it, sometimes the pregnant mother getting stem cell from the fetus into the bloodstream is getting her damaged organ to get fixed without any medical intervention. We have seen it when we studied the ethics. So it is because there is a surplus of stem cell available, and that damaged organ was easily able to get fixed. So that logic you should connect here with the abundance of stem cell, and especially when you see in aging and a disease condition. So scaffolds, when you want to define that, the scaffolds are artificial extracellular matrices. Extracellular matrix means a natural thing. If you can get it artificially, we have seen some example in the previous classes, many chemical names were there listed. Those who have doubt, you can go back and refer that. Artificial extracellular matrices that provide a three-dimensional structure for stem cells to grow and differentiate. So this is simple definition of scaffold. And the materials can vary from starting from natural scaffold to, because sometimes when you use some proteins or some material naturally occurring, but that is not an ECM. Say cellulose. Cellulose is not an ECM protein, but you can use it as a matrix or scaffold protein. Scaffolds can be made from various materials, including natural polymers such as like collagen. Collagen is an ECM protein, and synthetic polymers like polylactic co-glycolic acid, one example. Several examples we have discussed. And what are the functions the scaffolds can do to a stem cell? They can support stem cell adhesion and proliferation. They allow the cells to be attached and allow them to proliferate. We have seen it. Sometimes the synthetic polymers need to be modified with some amino acid derivatives so that the cells can find it comfortable to bind onto them. We have discussed that already. So the structure and composition of the scaffold can influence stem cell adhesion and growth. Their main purpose is that they will allow the adhesion and proliferation. They can mimic the niche microenvironment. So scaffolds can act as if the cell is at home. The cell do not feel I am in a strange place. It is making you feel, or the cell feel, homely. So they mimic the niche microenvironment. Scaffolds can be designed to mimic the physical and chemical cues a stem cell of present in a stem cell niche, influencing the stem cell's behavior. That means it should be able to make a copy of themselves, auto renewal, and it should be able to differentiate upon demand. They can promote differentiation. Scaffolds can be engineered to promote stem cell differentiation into specific cell types. If scaffolds are non-permissive, then whatever cues you provide, it will not get differentiated. So you don't want such kind of things to exist. And what are the applications? So scaffolds are used in tissue engineering to create new tissues and organs for transplantation. Simple. It's very simple. You want new tissues and organs for transplantation. And This can be of two types. One is wound healing, simple wound healing. Scaffolds can promote tissue regeneration and wound healing. Like if you have a severe wound, dermis got damaged, the burn wound, so you can put the scaffolds on the skin so that the newly formed stem cells can happily migrate with the help of the scaffold, and the skin healing become fast so that you will not end up getting too much of scar. Otherwise, deep wounds will give rise to too much of fibrosis and scar formation. And then comes the disease treatment. Scaffolds can be used to deliver stem cells for the treatment of several diseases. That many diseases, if it can happen because of, say, simple example, myocardial infarction. If infarct is nothing but a fibroid area what is present. So if you can remove them with some enzymatic treatment and allow some scaffold to be placed in that area where the fibroid or the collagen fibers are removed, and if the newly formed cells can migrate there, then you fixed the damaged area. Like that, heart I given as example, it can be liver, kidney, anything. Many other tissues can have similar kind of example. So now let us see stem cells in tissue engineering. I guess this slide we have discussed earlier also, but I am bringing it once again simply for refreshing your memory. Stem cells need to be isolated, they need to be characterized, they need to be expanded, they need to be differentiated. And for all these steps, except for isolation and characterization, expansion and differentiation, you need scaffold. So isolation, we know, it's isolate individual stem cell population, ensure that the cells retain their functionality and potential to differentiate, and characterize and track the stem cells from the different population, and ensure that the cells are healthy and powerful enough to get transplanted for expansion onto a scaffold, and culture the stem cell line in a stable multi or a pluripotent state, free from any mutations and of sufficient quantity and quality, and enable the economical expansion to make the cell therapy a reality. That means if the expansion involves 1 crore rupee, then people can't afford. So it should be so the choice of the scaffold and the strategy should be friendly enough for people to accept or adopt that technique. And then differentiation, control and activate the stem cell differentiation to the desired lineage. And functionally active differentiated cells can be used to put into the recipient. So what are the challenges in stem cell research? It is uncertain that the human embryonic stem cells in vitro can give rise to all different types of the adult body, because sometimes, although on paper yes it can, but in reality it may not. Say in a given tissue which you made, which has got 3 types of cell, A, B and C, stem cells can give rise to all A, B, C. Say if A and C is 25% and B is present 50%, then what if because of your differentiation B is now only 10% and A and C is more? Then it is not the real tissue. It has everything, but it's not the real tissue. So this logic is to be taken serious consideration. It is unknown if stem cells cultured in vitro, apart from the embryo itself, will function as the cells do when they are part of the developing embryo. You made a kidney tissue or heart tissue or liver tissue, but will they do as they are doing in an embryo or developing embryo? Stem cell development or proliferation must be controlled once placed into patients. You don't want the stem cells to continue to proliferate and become a tumor or an enlarged organ. Possibility of rejection of stem cell transplants as foreign tissues are very high. Although it is stem cell, but suddenly your immune system is detecting a large number of tissue and it may start attacking that tissue, and it can give rise to autoimmune cell. And contamination by viruses, bacteria, fungi, mycoplasma also is possible. So to summarize, in short, both niches and scaffolds are essential for regulating stem cell behavior, influencing their self-renewal, differentiation and migration. They are essential. Understanding these interactions between the stem cells and their niche is very important for developing effective stem cell therapies and regenerative medicine strategies. Now let us see what are the ways in which the cell scaffold interaction in reality, in practice, how do they interact. Let us see what are the types of interaction. Scaffold influence cell viability, growth function and motility. The types of cellular interactions under influence of scaffold are three types: adhesion, migration, and aggregation. So these are all the three outcomes of cell scaffold interaction. First one, adhesion. Most tissue derived cells require attachment to a solid surface for viability and growth. We know cancer cells can climb on each other, but whereas the normal cells need a substratum such as ECM or scaffold. Cell adhesion to a surface is critical because it is followed by other important phenomena like cell spreading, migration, and differentiated cell function. You also know when you find a place to stand, then you will think of sitting. If your leg is hurting, you will not think without standing. You will not think of sitting. You need a place to stand, then you sit. After sitting, you must think of stretching your leg. Same logic applies to the cells. It needs a scaffold, then it will think of spreading, proliferate, and spread. And the phenomena include cell attachment, cell spreading, and focal adhesion. So they are all the three main way in which the adhesion can take place. So techniques used to determine cell adhesion are sedimentation detachment assay, centrifugation assay, and fluid flow chambers. So cell attachment, the cells attach to the surface of the scaffold and form monolayer on the scaffold. This is what happens. And then comes the cell spreading. Surface attached cells divide and proliferate to cover the surface of the scaffold. The cells also penetrate inside the interconnected pores of the scaffold. That's why you have pores in the scaffold itself, so that it can form a intact tissue. Remember, later the scaffold get absorbed. It will be replaced by the ECM. And then comes the focal adhesion. Focal adhesions are large dynamic protein complexes through which the cytoskeleton, that is the protein present in the cell outside the cytoplasm, that is integrin, actin, and myosin, because the cell has a skeleton and at certain points the proteins will come out of the cell membrane and interact with outside. See, just like the pillars of a building are on the walls, but the pillars are not ending there, they are going into the basement and under the ground. Same logic. So if pillars are only there just touching the ground, the building, if one wind comes, it will fall down. So integrin, actin, myosin are such kind of proteins. And the cytoskeleton of a cell connects to the extracellular matrix, that is, in this case, it is called a scaffold. And this is a picture of different types of cells, different dimensions, and how they attach to the scaffold and they migrate. You can see some green fluorescent protein expression, etc. And then they migrate also, they proliferate and they migrate. So the techniques to determine cell adhesion: sedimentation detachment assay. So what they do here is sedimentation of cell onto a surface. You take a group of cells in a liquid medium, allow them to settle down. Incubation of the sedimented cells in the culture medium for some period of time, so that give them time to attach. Detachment of loosely adherent cells by removal of the culture medium by repeated washing. Just to decant it, whatever is not attached, it will go off. Whatever is healthy, it will attach and it will stay there. And the extent of adhesion is determined by the number of cells that remain associated with the surface or the number of cells that were extracted with washer. Say you had 100 cells you put into a tube and only 10 cells attached, 90 did not. Then you can tell 10% of the cells are adherent. You can know, and that is what you put it in here, allowed them to settle down and then washed. You see, you put so many cells and only here you can see only 6 cells attached of this many cells. So rest will wash away. And techniques to determine cell adhesion, another approach is centrifugation assay, the seeding of the cells onto the scaffold surface, that is incubation of the cells in culture medium for some period of time. And the plate is inverted and subjected to a controlled detachment force by centrifugation. Whatever we have done in the previous step, that has been done, and then you invert it. The extent of cell attachment is then quantified. So you made it, allowed them to attach, then you inverted it and then spun little bit, but attached cell will not fly away. Loosely bound or non-bound will fly away. So techniques to determine the cell adhesion also include fluid flow chambers. So here it's a simple technique. So fluid mechanical forces are utilized to produce a cell detachment in a well-controlled and quantifiable manner. So the cell suspension is injected into the chamber and the cells were permitted to settle down to the surface of the scaffold and adhere like you did in the previous two techniques. But first technique you just washed it, second technique you inverted and spun it. Here you don't do both. What you do is, after incubation, the fluid is forced between the two parallel plates and non-adherent cells are removed with the flow of the fluid. That means you are not decanting, you are not spinning, but you are laterally flowing the medium, and the unbound one will wash away, attached one will stay back, while the adherent cells remain on the surface, which can be quantified. So now comes the migration. So migration of individual cells within a tissue is critical for the formation of the architecture of the organ. Now we know, we assessed, okay, 10% of the cells are attaching or 5% of the cells are attaching, but are they attaching forever? Are they migrating? We need to have an assay. In tissue engineering, the ability of the cells to move in association with the scaffold surface or through the other cells will be an essential part of the new tissue formation or regeneration. If they move only, they can give rise to a proper tissue. So the techniques used to determine cell migration are under agarose test, under filter assaying, and under direct visualization. So the techniques to determine cell migration, the first one is agarose test. A cell suspension is placed in a well of the semi-solid agarose, which you cast a gel for running the gel, etc. Many of you would have done it. As you can see here, you put it, and the motile cells will crawl on the solid substrate underneath the agarose. There is a small gap. You put the cells here. If they are non-migrating, then they will stay here only after some time. If you watch it, maybe after an hour. But if they are migrating, they will go underneath. As you can see here, they will move. And motile cells will crawl underneath the agarose wherever there is a space. Then comes a filter assay. Cell suspension is placed on a filter with the small pores. As you can see here, tiny pores are there. You put it in here, the cells. The motile cells will crawl through the pores of the filter. They can move in material to the other side. They can crawl and go to the other side where they get detected. Just like somewhat like you have done this osmosis through a semi-permeable membrane, as you can see here. And the direct visualization assay, the paths of the movement of many individual cells are directly observed for cells migrating on the surface within a solid gels, where you watch it, take individual images multiple times. Every 10 minutes you take the image and then you superimpose, then you will see the cells are moving. Non-moving cells, dead cells, although they are attached, they will stay there itself. So then comes the aggregation. It is important in tissue development. So it is migrating now, attaching, migrating. But do they aggregate? That is the question. So it is important step in tissue development. It correlates cell-cell interaction with during cell differentiation. The viability and migration for subsequent tissue formation, the aggregation property is very important. Aggregate morphology allow re-establishment of cell-cell contact in tissues. Thus, the cell function and survival rate are enhanced in aggregate culture. If cell is moving but it's not forming aggregate, it doesn't help much in forming a organ. The formation of the aggregates by incubating cells in suspension and adding serum proteins to promote cell aggregation. This is one approach. So the techniques used to determine aggregation are direct visualization, electronic particle counter, and aggregometers. So these are all the approaches used in measuring the aggregation properties. So the direct visualization, that is monitoring the aggregate size to determine the extent of aggregation. So just to monitor them, watch it, how big it is becoming. And electronic particle counter, so that is invented by Muscona, determines the kinetics of aggregation by measuring the aggregate size distribution over time. So this is the approach done through electronic particle counter. This procedure utilizes the computer image analysis to follow disappearance of a single cell within a given time. You can use electronics devices to measure that. Aggregometers are simple. They are small angle light scattering through the rotating sample cuvettes are used to produce continuous record of the aggregate growth, just like how you measure the bacterial growth through a cuvette. So you can, aggregometers can be used whether they are joined together, it will reflect in their absorbance spectrum. So we will study more about the regeneration biology in the next class. Thank you.